Hi, Advanced Cell. What I wanted to do here is give you a little preview to what's to come in our RAB review article. So this background information I think is going to make the review article a little easier to follow. So after the stage is set. So the major question that we're going to ask in this week is how do things get to where they need to go? So for example, we just talked about how vesicles bud off of the ER by building COP1 coats. Now that they pinch off after that budding event, how do they know to go to the ergic? Or how do they know to go to the Golgi? How do they know to go to the plasma membrane to get secreted? So the COP components like Sec23, 24, Sec31, and 13, they help to build the coat, but they don't actually have the signposts or the zip code to tell that vesicle where to go. So it turns out that there's proteins called Rab proteins, and these are some of the master regulators for regulating intracellular trafficking, telling these vesicles where they should go. So let's take a step back and look at Rabs in general. What are Rab proteins? So the Rabs belong to a superfamily of GTPase molecules. Okay, so GTPases hydrolyze GTP to GDP, and they regulate a lot of different biochemical processes. So examples where you may have come across these before are in our own class. We learned about how the signal recognition particle and SRP receptor are G proteins. They require GTP hydrolysis in order to separate and to go on and do their business. So we also, maybe in 41C, talked about tubulin. Tubulin is also a GTPase in the sense that the tubulin subunits have GTP that gets hydrolyzed to GDP. Other things like initiation and elongation factors for translation. Some other things you might have heard about are the heterotrimeric G proteins. So these are involved in G protein coupled receptor signaling. And so things like G alpha, G beta, G gamma that are activated by G protein coupled receptors also are G proteins. And then finally, what we're going to talk about today are the RAS superfamily, which are small GTPases, very small molecules, of which the Rabs are one family under this bigger superfamily classification. So what do the Rabs do? Why are we bothering spending a whole week just on one class of proteins? Okay, and it's because Rabs do so many things. They're essential for membrane trafficking. They do a lot of other things as well, but we're going to concentrate on their role in membrane trafficking this week. So the review article that you're going to read has a lot of different details, and I don't want you to focus on the nitty-gritty details about what individual Rabs do or the very specific proteins that are involved, but instead I want you to focus on these big ideas. So taking away from the reading, how are Rabs activated and inactivated? So this is called the Rabs cycle. How do they cycle on and off membranes? What instructs them to insert into the membrane and to come off again when they're not needed anymore? What do the Rabs do? Or maybe what I should really ask is what do they not do? Because they seem to be involved in everything. And then finally, how is their specificity regulated? There's tons and tons of different Rabs, as you can see from this figure. If you look in, so here's the nucleus, and here's the ER, and the Golgi, you can see all these different vesicles, all these different compartments have different Rab proteins on them. There are over 60 different Rab proteins in humans. So what do each of them do, and kind of what's their role? So the main role that we're going to concentrate on this week is their role in defining membrane compartments. So another way to think of this is that they're molecular signposts. They tell our vesicles where to go. They tell our membranes of the endomembrane system what they are. Okay, so as I mentioned, mammals have over 60 Rabs, yeast have about 11 of these, and so these different Rab proteins are localized to different compartments within the secretory pathway, and they help to define membranes. So for example, for ER to Golgi vesicle transport, proteins called Rab1 are important for this step. To go from the Golgi to the plasma membrane, there's a Rab called Sec4 that's involved in this. Or even for things like we're going to talk about later in the course about endocytosis and endosome sorting, Rabs are essential here too. Some of the famous ones are Rab5 and Rab7, which we're going to talk about in class. And again, I don't want you to concentrate on the specific Rabs, but more generally, what is it that's defining these different membranes? How does an endosome know it's an endosome? How does a Golgi know it's a Golgi, right? And Rabs can be some of those signposts that tell these membranes what they are, but they also can help send vesicles to target membranes by having those Rab molecular signposts on them too. So let's take a closer look at what Rab actually looks like. So here is the structure of Rab6A. And we can see GTP here in its active site. And so the combination of a domain called switch1, here shown in dark blue, and switch 2, shown in light blue, along with something called the P loop, and that's P stands for phosphate. The combination of switch 1, switch 2, and the P loop help to set up the binding site for GTP. And this is the active site of the enzyme itself, right? Because Rabs are G proteins, and G proteins are defined as having GTP-ACE activity, meaning they can hydrolyze GTP. So this is where their enzymatic active site is. They have a different domain that's important for binding things called effectors. And the effectors are things that bind active Rabs and tell that Rab what to do. So let's take a look at this step of GTP hydrolysis, since it's so important for Rab's function in defining whether a Rab is turned on and active or turned off and inactive. So this is kind of a little GIF back and forth that'll show you the difference between an activated and inactivated G protein. It's actually RAS, but the Rab has a very similar mechanism. So here we can see the switch 1 region and the switch 2 region, and here we have GDP. There's the guanine, and then there's 1,2 phosphates. And this GIF is going to toggle back and forth to show what happens in the presence of GDP versus GTP. Okay, so if I start it, there's GTP. You can see with the GTP, the switch 1 region changes conformation and lifts up. The switch 2 region undergoes some slight changes, and what that does is it helps to define whether the Rab protein is going to be active or able to bind things called effectors, or whether it's going to be in its GDP state and inactive and unable to bind its effectors. So the term effectors just means things that are going to bind that protein that carry out the business end. So the Rab is kind of like the light switch, meaning that you turn the light on and light illuminates the room, right? You turn the light off, now the room is dark. Same thing here. You turn the light on by adding a GTP. Now the Rab can illuminate the area by having effectors bind it and carrying out different processes. Okay, so what is it that allows this GTP hydrolysis to take place? Because that's such an important part of its function. So we mentioned in class about the role of the different G protein regulating proteins called GEFs and GAPs. So a GAP is a GTPase activating protein, and that means it's going to activate the GTPase domain in order to cleave GTP to GDP. So let's take a closer look at how the GAP actually works. So in light blue here, cyan, this is going to be our Rab G protein. And then in kind of the more green color here, this is going to be the GAP, or the GTPase activating protein. So this is the enzyme that catalyzes GTP hydrolysis in Rab. There's something in the GAP called an arginine finger. So there's this extension of the protein that inserts into the active site of our G protein, or Rab. And one of the most important catalytic sites there is made up of this arginine finger. So arginine, the arginine residue sits in here, and it helps to stabilize the beta phosphate of GTP. So that allows the terminal phosphate to be hydrolyzed, and yet that molecule will still be stabilized. Okay, so here's the arginine from the GAP. It helps coordinate here this glutamine residue in Rab, and that can help with the hydrolysis as well. Okay, so this active site of Rabs is really important because we can actually manipulate it experimentally in order to study active versus inactive versions of the Rab proteins, or any G protein. So again, I mentioned the important thing here. There's a glutamine in Rab, which can help to coordinate the arginine finger here for the GAP. So if there's mutations in this glutamine in the Rab, we can lock our Rab into a GTP bound state, because now our arginine finger cannot come in and interact and be a catalyst. Likewise, there's a serine right here, where if we make mutations to that serine, it can help lock Rab into a GDP bound state. So these tools, these mutagenesis experiments, have been essential in understanding how GTP and GDP play roles in the functioning of Rab proteins. So let's take a closer look now at this idea of hydrolysis and what hydrolysis means for these proteins. So this is kind of a complicated slide at first, but we're just going to take it step by step, going from step one all the way through to the end. Okay, so when a new Rab protein is born, basically when the ribosome translates a new Rab protein, GDP is going to be in its site. Okay, in its active site, there's a molecule called the REP, which is a Rab escort protein. I don't have the definition here, but it's an escort protein, and what it does is it brings Rab to some enzymes called geranyl transferases. Okay, it's a mouthful, but basically what these enzymes do is they insert geranyl groups into Rab, which are long hydrophobic molecules. Okay, they're like kind of long lipidy molecules, and so it modifies the Rab with these geranyl groups. and those are going to be what later insert into the membrane because they're hydrophobic. Okay, so once the geranyl-geranyl modifications are added, the Rab escort protein can escort or bring Rab to its site of action. Okay, so it's escorting it for the very first time to a donor membrane where there's a signal for the Rab to come to this membrane. And often these signals are going to be read as the different types of phospholipids that are here. So different Rab proteins can recognize different phospholipids. All right, so escort protein brings Rab to the membrane. The geranyl-geranyl hydrophobic groups allow it to insert into the donor membrane, and while it's hanging out there, it can encounter something called a GEF, which is the guanine nucleotide exchange factor. So this is the activating protein, the activating enzyme that takes GDP out of the active site and allows GTP to flow in. Okay, so it doesn't actually place GTP in. There's a high concentration of GTP in the cytoplasm, and so the GEF actually removes GDP, and then just the existing GTP allows the replacement of the active version here in Rab. So once Rab is activated by GTP, now we've gone through that conformational change that we saw up here, and so now the Rab is capable of binding what are called effectors, or the things that do the business. Okay, so now we've got active Rab and its effector. This can be a trigger or a signal for vesicle budding or for transport along the cytoskeleton like microtubules or actin. Okay, and it's this Rab interacting with this effector that'll tell this vesicle to go, for example, from the ER to the Golgi or to the Urgic. Okay, so once this vesicle binds the acceptor membrane, now the Rab is done with its job, and so the complex will dissociate. The Rab is now going to be in its GTP form, but we don't need it to be active anymore. Now the enzyme called the GAP will come in, and again, this GAP is what hydrolyzes GTP to GDP. Now Rab is in its GDP form, and when it's in that form, it does not actually like to be associated with the membrane. Okay, it's less stable. It's much more stable when it's in its GTP form associated with the membrane. So what happens is the protein's called GDIs, or GDP dissociation inhibitor down here. GDIs will come and interact with the GDP-bound Rab, extract it from the membrane, and then remember we've got these geranyl-geranyl groups. GDI helps shield those hydrophobic groups while it's in the cytoplasm, and the GDI helps bring Rab back to its original donor membrane where it began its kind of life story here. So the GDI, and this is what the Cheney lab works on, the GDI can escort or remove the GDP-bound Rab from its final membrane, return it back to its original membrane, so then it's primed and ready to go and able to be activated by a GEF again to be activated Rab and complete this cycle all over again. So this is the life cycle of Rab, is that it transitions from an inactive GDP-bound state, becomes activated by the GEF proteins, and then can bind effectors to do its job. Once its job is done, there's GAP proteins that turn it off by hydrolyzing GTP to GDP, and then the GDIs will come and extract it and recycle it back to its original membrane to do its job all over again. Okay, so there are a lot of different aspects of trafficking that require the Rab proteins, and the reviews will go into some of this. So, for example, it's important for sorting. It's important for sorting some cargo proteins. It's important for recruiting code components. It's also important for uncoding the code once it's pinched off from, for instance, the ER. Rabs can help to recruit motor proteins to allow the vesicle to move along microtubule motors. It's important for tethering, so interacting with things like tethers that bring a vesicle close to a donor membrane so that the snares can fuse and actually do the membrane fusion event. So all of these steps in vesicle trafficking, the Rabs can help to regulate. Again, it's probably the most important protein you've never heard of before. So I wanted to return to this idea of what's going to be in the review article. Just to remind you, Rabs do lots of things. There's going to be a lot of details in the reading. Focus on these big ideas. How are they activated and inactivated? How do they get on and off membranes? What do they do? And then particularly important for us, we're going to spend some time in class, is how is that specificity regulated? So you can start to pay attention to things in the review article like the importance of Rab microdomains in determining membrane identity, the importance of phospholipids in this process, the different phosphorylation events that happen on phospholipids can define membranes as well, and Rabs are essential for setting this up. There's something called a Rab cascade. There's also something called Rab conversion. So these are kind of the next step topics once you understand the basics that we've just talked about with Rabs. These are kind of taking it to the next level to figure out how they're actually completing these very important processes in the cell. So I'm going to leave it there and let you carry on with the review article, and we will see you next week.